Assalamualaikum. I am Arya Jamil, coming from University of Agriculture, Faisalabad, and currently I completed my PhD from University of Agriculture, Faisalabad in veterinary pathology. And now working as research associate in Cinder UF project. And today in this international seminar on the poultry health management, the topic of my presentation is Bacillus Licheniformis for sustainable poultry health. First of all, introduction of the mycotoxin. Mycotoxin, these are secondary fungal metabolites produced by various toxigenic fungi, especially belonging to the genus Aspergillus, Penicillium, and Fusarium. And according to the FAO, 25% of the bird crop, these are contaminated every year due to the mycotoxin. And according to the mycotoxin survey, highly prevalent mycotoxin in South Asia is aflatoxin and followed by the ochratoxin. And this sketch represents you that how mycotoxin it affects the gastrointestinal tract and act as a predisposing factor for necrotic enteritis. As by the ingestion of mycotoxin contaminated feed, the first organ that is exposed to the mycotoxin, it is the gut. So mycotoxin, it has this very toxic effect on the gut, and mycotoxin, it decreases the gut integrity, gut immunity, and gut barrier function. And this can lead to the dysbiosis. And mycotoxin, it also lead to decrease the nutrient absorption from the gut and create the ideal condition for proliferation of the Clostridium perfringens, especially in the small intestine due to the alkaline pH. And now about the Bacillus licheniformis. It is Bacillus licheniformis. It is gram-positive spore-forming facultative anaerobe, and it is rod-shaped and motile bacteria. It having the antimicrobial and antioxidant effect as it squelch the free radicals that produced by the mycotoxin and alleviate the oxidative stress. And it also biotransforms the mycotoxin from the highly toxic metabolite to less toxic metabolite. And it having the antimicrobial effect as it balances the normal gut microbiota and also enhance the gut integrity, gut immunity, and protect the gut barrier function by enhancing the beneficial gut microbiota and improve the performance of the bird. Now about the gap analysis. Despite the extensive research in the literature, significant gap persists concerning to the confirmed effect of mycotoxin and necrotic enteritis. And no comprehensive information it is available about the protective effect of Bacillus licheniformis in broiler bird exposed to mycotoxin and necrotic enteritis. And the main objective of this research work was to determine the role of mycotoxin in aggravating the pathological changes induced by the necrotic enteritis in broiler bird, and to assess the beneficial impact of Bacillus licheniformis in broiler bird exposed to mycotoxin and necrotic enteritis individually and concurrently. And now about the statistical analysis. Data obtained from the experiment was subjective to the analysis of variant tests, and Tucky test means were compared by the Tucky test to elaborate the significance of alteration in different parameter studies. Now about the methodology. This was the plan of the study. Before the in vivo studies, such as production and quantification of mycotoxin, both upland ochratoxin, and the isolation and molecular detection of the Clostridium perfringens and propagation of the Bacillus licheniformis. First of all, production of the mycotoxin. For the production of the aflatoxin, short-well method was followed, and rice was used as the substrate. And for the production of the ochratoxin, Trank et al. Method was followed, and wheat was used as a substrate. And the pure culture of Aspergillus flavus and Aspergillus ochraceus, it was imported from the Spain and propagated on the potato destrose agar, and then further inoculated on the wheat and rice for the production of both upland and ochratoxin respectively. For the isolation and confirmation of the Clostridium perfringens, sample was collected from the clinically suspected bird, and sample was collected from the small intestine, especially jejunum and the ileum, and then sample was pre-enriched in the thioglycolate broth, and then inoculated on the specific media that is tryptol sulfide cyclosidine agar, and then incubated anaerobically for 24 hours at 37 degrees centigrade. Then further confirmation was done by the Grandstein, Schiebler, Decker, and molecular identification was done. This was the few glimpses about the isolation of Clostridium perfringens in the toxicological lab of Professor Dr. Kashif Snemi. And now in this slide, enlisted all the primer that was used for the confirmation of Clostridium perfringens and enlisted the complete profile of the PCR. Now about the propagation of Bacillus Licheniformis. Pure culture of the Bacillus Licheniformis, it was imported from the Thailand, TISTR, Thailand Institute of Science and Technology Research. And this was the few glimpses for the propagation of Bacillus Licheniformis on the nutrient broth. Now about the layout of the experiment. For this experiment, total 280 broiler birds were procured from the commercial hatchery and divided into the 8 groups, each group having the 35 birds. Group A was control negative, Group B was supplemented with Bacillus Licheniformis, Group C and D was challenged with mycotoxin and Clostridium perfringens infection respectively, and Group E was co-challenged with mycotoxin and Clostridium perfringens infection. And Group F and G was challenged with mycotoxin and Clostridium perfringens infection respectively, and supplemented with Bacillus Licheniformis. And Group H was challenged with mycotoxin and Clostridium perfringens infection and supplemented with Bacillus Licheniformis. After 2 days of the age, birds were offered mycotoxin contaminated feed and Bacillus Licheniformis, and necrotic enteritis was induced by the oral watch method at day 17, 18, and 19 day of the age. And the duration of the experiment was 35 days. Now about the parameters that were studied during this experiment, which included the production performance, such as body weight, feed intake, FCR, and the relative organ weight, and the serum biomarker, which include the hepatic and renal biomarkers, and the hematology will include the rethogram and the leukogram, and the gross and microscopic pathology of the liver and the intestine. Now about the result. Image A showed the growth of the Clostridium perfringens on the TSC agar, black colony appearance on the TSC agar. And the image B showed the double zone of hemolysis on the sheep blood agar. And the image C showed the ground staining under the microscope, rod-shaped bacteria was observed. And image D showed the molecular identification of the Clostridium perfringens. 279 base pairs show the species-specific identification of the Clostridium perfringens, and then further confirmation was done by using the specific gene, such as 324 base pairs show the alpha toxin production, and the 196 base pairs show the beta toxin production, and next band show the multiplex PCR. This image shows the growth of the Bacillus Licheniformis on the nutrient agar. And the CFU count per ml was 2.5 into 10 to the power 7 CFU per ml. And now about the person mortality. During the experiment, highest mortality was observed in co-infected group E, followed by the group H, and then the individually infected group C and D as compared to the control negative group A, and all the remaining group minor mortality was observed throughout the experiment. Now about the weekly body weight. During the week 1 and 2, body weight of all the group was non-significantly different from the control negative group A. And while during the 3rd, 4th, and 5th week, body weight in co-infected group E was significantly decreased, followed by the group H and then individually infected group C and D as compared to the control negative group A. Now about the feed intake. During the 2nd week, feed intake in group C and E was significantly decreased as compared to the control negative group A. And during the 3rd, 4th, and 5th week, feed intake in group co-infected group E was significantly decreased, followed by the group H and individually infected group C and D. Now about the FCR. FCR was significantly highest in co-infected group E, followed by the group H, and then individually infected group C and D as compared to the control negative group A. And now about the relative weight of the liver and kidney. Relative weight of the liver and kidney was significantly highest in co-infected group E, followed by the group H, and then individually in petty group C and D. And while the intestinal weight was significantly highest in co-infected group E, followed by the individually Clostridium infected group D, as compared to the control negative group A. Now about the hematology. Erythrocyte count was significantly decreased in co-infected group A, E, as compared to the control negative group A, while the leukocyte count was significantly highest in Clostridium perfringens infected group D, and lower in mycotoxin infected group C, as compared to the control negative group A, while the hemoglobin and PET cell volume was significantly decreased in co-infected group E, followed by the group H, and then individually infected group C and D, as compared to the control negative group A. Now about the serum biochemistry, hepatic biomarker ALT and AST value was significantly highest in co-infected group E, followed by the group H, and then individually infected group C and D, as compared to the control negative group A. And the similar pattern was observed in the renal biomarker. And now about serum protein, total protein and albumin value was significantly decreased in co-infected group E, while the globulin value was significantly highest in Clostridium perfringens infected group D. Now about the gross and microscopic pathology of the liver. Images show the liver of the control negative group A, while image C show the co-infected group E. You can see here the severity of the infection. In co-infected group E, glossy liver appears darkish to blackish appearance and pinpoint hemorrhages. Also, the severe changes microscopically severe hemorrhages and the evacuation was seen. While in the evaluated group, this was comparable with the control negative group A. Now about the gross and microscopic pathology of the intestine. Images show the intestine of the control negative group A, while the image B show the intestine of the Clostridium perfringens infected group B, while the image C show the co-infected group B. You can see here the severity of the infection in co-infected group B, disruption of the villi, loss of the goblet cell, while the ameliorated group D, you can see here, this was comparable with the control negative group A with the slight loss of the target. On the basis of this result, it can be concluded that mycotoxin increase the severity of the necrotic enteritis and serve as a predisposing factor for necrotic enteritis. And the supplementation of the Bacillus Licheniformis as the dose rate of 1 into 10 to the power 6 CFU per kg feed ameliorated the individual pathological alteration induced by the mycotoxin and necrotic enteritis, while partially ameliorated the concurrent effect of mycotoxin and necrotic enteritis. And now about the reference. And at the end, I acknowledge these two persons, Professor Dr. Mohammad Kashif Slimi and Dr. Aisha Khatun. Actually, today I am presenting in front of you due to the blessing of my Allah and the hard work and the prayers of my parents. And also due to the efforts, because actually I completed all my MPhil and PhD research work in toxicopathological lab of Professor Dr. Kashif Slimi.